So I have a few slides to go through DNA mutation. So what happens when the plasmid DNA from these plasmids, broken up or intact, gets into the nucleus of the cell? Well, you can get DNA mutation, and that can occur through substitutions, through deletions, and in this manner, I'm going to go through a number of ways that the plasmid DNA, and even without the plasmid DNA, that these shots could cause cancer. So the 9 potential ways to induce cancer or more. One, the lipid nanoparticles themselves can take mRNA and DNA to all cells, and they've been shown to readily transfect hematopoietic stem cells. LNPs have also been found to cause cancer cells that are already present to more readily spread by inducing endothelial leakiness. There may also be an oncogenic effect of the LNPs themselves, which has not yet been studied. As Kevin said, there are SV40 elements to the plasmids. This is extremely concerning, particularly because they were not disclosed to regulators. So the SV40 promoter is very promiscuous. It's a super promoter. It's great at driving gene expression. And if that should sit above an oncogene, of course, you could have an explosion of an amplification in a cancer gene. The SV40 enhancer region, the nuclear targeting sequence, as Kevin described, also takes the DNA to the nucleus within a very short time period. It is designed to do that so that you get effective gene therapy, insertional gene therapy. So the spike protein itself can also interact with and inhibit the tumor suppressor protein p53. That was shown pretty early on. And then plasmid DNA does not need to have the SV40 sequences in order to be able to cause insertional mutagenesis and to go to the nucleus. There are lots of proteins that assist in carrying and binding to and carrying that exogenous DNA to the nucleus, where it can then be integrated. So insertional mutagenesis can cause something called frameshift mutations, which also lead to aberrant proteins being made. Those aberrant proteins can also lead to cancer. mRNA itself can be reverse transcribed to DNA and then also integrate in the genome, which causes cancers. And this is particularly true in the ovaries and the testes, where line 1 is more reverse transcriptase is more constitutively expressed. So that's a real concern there. RNA, through a mechanism that I'll go through coming up, can also be reverse transcribed to DNA, and then that DNA back to RNA, and then to cDNA, and then be passed on. There's another mechanism through which these could cause cancer, and that is through immunosuppression of T cells, particularly T cells that keep cancer from expanding in these stochastic niches where they guard the cancer clone and keep it from expanding. We see this in our pets as they grow older, that once we have immunosenescence and thymic involution, then you see an explosion in these sarcomas and lipomas and other cancers because of this, these T cells not being present to stop clonal expansion. So there are different types of genetic mutations. There's somatic mutations, which only affect the cells outside of the gametes. And then there's germline mutations, which affect the gametes. Now, here it says that a somatic mutation cannot result in a hereditary passing on, but there is a mechanism through which you can have extra chromosomally passed genetic elements be passed through sperm. And it is a very interesting mechanism of epigenetic regulation. and that is called sperm-mediated gene transfer. I'm trying to make sure I don't go over here. So ways to pass on genetic vaccines to progeny through both male and female germ cells. I spoke a little bit about this in December of 2022 at the U.S. Senate. My very large concern that these gene therapies will be passed on to our progeny and will contaminate the gene pool. And this is not being investigated at all. Not a single person has investigated sperm or ova to see if these are being genetically integrated. And I have reached out to multiple labs asking them if they would investigate this. We have an in vitro lab that is willing to work with anyone who is willing to test both sperm and ova for integration. So in the first, you can have integration into the genome directly of gametes from the DNA-based vaccines, or through reverse transcription of RNA into the coding DNA. And of course, the DNA plasmid sequences then make that possible as well. We know that these go to the testes and ovaries, and we know that they can be taken up there and integrated into the gametes. Genomic integration could result in cancers rather than just functional integration. In fact, it's unlikely that we will have functional integration into the genome, creating a spike protein, but more likely that you'll have insertional mutagenesis leading to cancer. But there is another mechanism which you can pass on functionally expressed spike protein, and that would be through this extra chromosomal DNA or RNA through sperm-mediated gene transfer. So both of these mechanisms could result in a constitutive expression of the spike protein in some or all cells of the progeny, depending on the way it's inherited. But again, less likely through actual integration into the genome, more likely through this mechanism called sperm-mediated gene transfer. So oocytes can also pick up exosomes or LNPs, and of course, the DNA can be incorporated into the genome. So this is a picture of some sperm that have been transfected with exosomes carrying DNA, and this is courtesy of Dr. Corrado Spadafora, who really was the first to show that sperm-mediated gene transfer was occurring. Through this mechanism, we could be passing on these genetic elements to our children. Sperm-mediated gene transfer shows that it is not required to have stable integration into the genome in order to pass on low copy number, chimeric expression of extra chromosomal DNA expression. And this is very concerning. So this can pass on to 2 generations in this manner. And it's something that most definitely must be looked at along with the actual integration of the DNA itself. So thank you very much. I will end here. I believe my time is up, and thank you so much for allowing me to speak.